Good morning and good afternoon, everyone. Welcome to the last day of the workshop. So I will just go over some general information, then go through the presentation. Please use the question answer panel for asking questions. And if you would like to speak, please raise your hand so that we can unmute you. So today's plan is that after a demonstration of the sum of tools that we have already seen, attendees are welcome to share any results or experiences that they got by exploring some of the tools over the past 3 days. As I go through the demonstration, our colleague will post the respective links in the chat so that as I am going through the demonstration, you can try those. And if you face any questions, please raise hands so that panelists can unmute you and intervene me so that if there are any questions, I can answer them. And of course, please do provide feedback following the Google Forms link shown here. It is also listed in the details web page above the table for the detailed schedule. And of course, there are various other modes through which you can provide feedback or raise issues and ask questions related to these tools. So today we are going to use various tools to explore and learn more about human liver cancer. So I will do a short demonstration of these tools in the context of human liver cancer. So as an example workflow, a researcher may be interested in finding what are the mechanisms and drivers of hepatocellular carcinoma in humans. So one would generally start with some literature search from which one may find a list of key genes or metabolites related to hepatocellular carcinoma. And then with those starting list of genes or metabolites, one could take several routes. For example, one could go and search on the Metabolomics/Workbench website based on species such as Homo sapiens or human, or anatomy such as liver in this case, and cancer as the disease. Or if you have a list of genes to begin with, then you can go to MedGene and find what are the related reactions, metabolites, and studies in Metabolomics/Workbench, which are relevant for those genes. And of course, at the CFDE portal, potentially, we can find information on the study on the disease of our interest from other resources, such as other data coordinating centers. So today, I will begin with given a list of genes, apply the MedGene analysis to identify what are the studies in Metabolomics/Workbench, and then show how various tools can be applied on those analyses, such as t-test, clustering, classification using OPLSTA, and METNP. Then we can visualize the results from these, applying these methods and interpret the results to potentially generate novel hypotheses or validate some of the results that we know from the literature. So from the literature search, one can get a list of genes that are implicated in hepatocellular carcinoma. So for example, in this case, we found 4 genes, and of course, there can be many other genes which might be relevant. The first is pyruvate kinase, which is significantly upregulated in hepatocellular carcinoma. It's involved in tumor growth, and it's associated with poor diagnosis, poor prognosis of the disease. And these are the papers which highlight about this finding. Then the gene methionine adenosine transferase 2A is involved in the regulation of S-adenosine methionine, or SAM, and results in dysregulation of methionine metabolism. And it's also a marker for progression and prognosis of hepatocellular carcinoma. We have some papers which talk about that gene. Then we have the gene ACSL3, or acyl coenzyme synthetase long chain family member 3, which along with the gene MAT2A forms a subpathway that protects cells from ferroptosis, which is a type of cell death. This pathway is particularly active in hepatocellular carcinoma, helping cancer cells survive under metabolic stress. So essentially, it contributes to the survival of cancer cells and then tumor growth. There are several papers which talk about the prominent role of this gene in hepatocellular carcinoma. Sorry, that link got clicked. So we go to MedGene, and at this point, please post the link for MedGene in the chat so that the attendees can try it themselves. In the gene ID box here, you can type the gene ID, gene symbol for the 4 genes separated by comma, and select the symbol as the type. Or sometimes it might help to select a symbol or alias for the gene, in case some of them could be gene alias instead of the official gene symbol. Then in the organism menu, you select human as the species. For anatomy, you select liver. Then for the disease or phenotype, first you select neoplastic disorder as the higher level category. And then in the lower pull down menu, you select cancer as the disease. And then click on the submit button. Then you get the main results page where you see a schematic of showing how the results that we are presented, how they are derived. For example, first you search for the gene, then you find out in which pathways that gene is involved, what are the reactions associated with that, and then what are the metabolites which are participating in those reactions. And finally, what are the studies which have measurements of those metabolites. I'm going to pause for a brief second in case I see some posts in the chat. In case there are any questions, please ask and then I can answer. Okay, I will proceed. So, because we are interested in finding what are the studies at Metabolomics Workbench relevant to the genes and metabolites, we go to the studies panel, and here we see there are 2 studies which are showing up for many of these metabolites. The first is the study 230, ST000230, and 231. So now that we know some studies that we can focus on, we go to Metabolomics Workbench following those links there. And here we have the summary page of the study 230 and 231. I'm going to focus further on analyzing the data from study 230, just to illustrate. I will wait just for a few seconds here so that please post the link in the chat for visiting the page for this study, so that attendees can, if they wish, they can try to go through these tools and analyze the data from this study. So I'm going to look at a couple of analysis, and of course there are a number of other analysis tools available, but just for illustration purposes, for example, we want to see what is the distribution of the metabolite counts with respect to different metabolite classes as a pie chart. Then we will look at the volcano plot analysis. So from the pie chart at the level of main classes, we can see that a majority of the metabolites belong to the amino acids and peptides group. And then the next, besides the others, then we have the purines and then fatty acids and monosaccharides, etc. Then we looked at the volcano plot analysis, where let me briefly walk through how you set up this panel here. So here, essentially, it is going to perform a t-test between 2 conditions. So you need to select which factors what are the factor values you are going to include in the 2 groups. For example, in this case, in the first group, we include samples corresponding to hepatocellular carcinoma. And in the second group, we include samples corresponding to surrounding the non-tumor tissue next to the hepatocellular carcinoma sites. Then you can select the values for these parameters, such as p-value cutoff. And the fold chain cutoff is one, essentially, that is there is no filter applied with respect to fold chains. Then one can choose the sample normalization method such as mean, sum value, or none, no normalization applied. Then you can look at the classification with respect to subclass, main class, or superclass. And for displaying the names, you can use either reference name or the names submitted by the metadata submitter. Then here we have the, if a study has more than one analysis, for example, in this case, we have ESI negative ion mode and ESI positive ion mode, then we have the option to select all of them. So sometimes that might be the best option. So that way you can get the results from all the metabolites measured. Then you click on the run volcano plot button. And after a few seconds, you will see a panel of these results here, which I will go through. So in the volcano plot, we right away see that S-adenosylmethionine, or SAM, is strongly downregulated in non-HCC versus HCC, or we could interpret it that it is strongly upregulated in HCC versus non-HCC condition. We can also look at these results at the metabolite class level as a bubble plot, as it is shown here on the right lower side. Then we have two more sets of results. One is looking at these results as a bar plot of significant metabolites organized by at the level of metabolite classes here. And you can see different classes here: amino acids, dipeptides, etc. And then we can see here in the Kolmogorov is not test based and class enrichment that amino acids are prominently enriched, followed by nicotinic acid alkaloids. Then the next analysis we looked at is looking at clustering of data from these two analysis conditions separately: ESI negative ion mode and ESI positive ion mode. In each case, we can see there are one or more clusters of metabolites which are changing together. And we can further analyze them based on like what functions they might be related to. Then we looked at some classification history tools, such as OPLS discriminant analysis. Here you select two groups and then run the analysis. And as we can see here, in this case, we are seeing a clear separation between the two groups based on OPLSTA. And the plot on the left bottom side here shows the VIP score, which essentially tells that what are the metabolites which are contributing significantly to the classification performance. So that is quite a good number of tools already available at the Metabolomics Workbench. But if the user is aware of other tools that are out there in the literature, or they have developed their own custom tool, they can download the actual data files and perform any additional analysis and interpretation. So besides these tools, we also applied the METNP pipeline on this, where we selected data from both the modes, and then we select the tissue type as the factor with respect to which we will be doing the t-test. So I'm going to pause here for a little bit so that if someone is interested, they can follow the link posted in the chat. So panelists, if not already, please post the link for applying METNP, either the ones which are shown here. So if you go to the stats toolbox page, usually you will see as in that for this study, you will see a link for applying METNP. And that is the link, the second link shown here. So one thing I must say that applying METNP on this study can take longer than 5 minutes in some cases. So just something to keep in mind, that it is possible that the analysis may not complete when many people try it at the same time. So the results that we have here are essentially these 6 plots and corresponding tables, so that one can do any further analysis with those results that we see here. For example, we have the class enrichment at the subclass level. Then we have the volcano plot, and again, as we saw earlier, SAM or S-adenosylmethionine is strongly downregulated in the non-HCC versus HCC comparison. Then on the right-hand side, we can see that this metabolite adenosylmethionine is participating in many, many pathways, highlighting the central role of this metabolite in terms of being able to potentially affect various pathways and functions. So these set of results provide a good starting point to even continue further exploration towards interpretation and mechanistic understanding of what is happening in this disease. And these are some of the metabolites, and as we saw those 4 genes, one can further follow upon them with respect to network-based analysis. So with that, while that was still a large set of results that we could see, there may be more information out there related to this disease and these genes or metabolites that we saw. So we decided to go to the CFDE data portal and search for, based on certain keywords related to the disease and these molecules, and see what we get. So for example, in this case, we looked at this key metabolite that we saw based on several analyses. And when you search for S-adenosylmethionine, we get several records from the data coordinating center Glygen, which has information on glycosylation of proteins. So that means that the information that is coming up from Glygen is potentially related to the glycosylation of enzyme corresponding to the reaction in which this metabolite S-adenosylmethionine participates. So I'm going to pause here just for a little bit so that attendees can potentially go to the website and try this search themselves. And panelists, please post the link in the chat if not already. On this page, the results page, you can click on in the view column on the headline sign to find more details about that particular record, such as if it is associated with that project. There may be a project description, at least in some cases, and then other information related to that specific data set, such as what is which protein is being glycosylated in this case, and additional information. If there are any files associated with this record, then you may have the ability to download those files and look at any further details. Moving forward, we also search for liver Homo sapiens cancer on the CFDE data portal. And it came up with a number of results. Actually, in this case, there are more than 20,000 records here coming from several DCCs such as MetabolomicsWorkbench, Glygen, xRNA, which stands for Extracellular RNA Consortium. And we also have the results from Links DCC, which is not showing in the top 10 here, but nevertheless, we know that it is there. So it might be difficult to go through this many results, so we would want to further narrow it down. So we apply filters for anatomy, species, and disease as shown here. So you can go to the right-hand side filter menu here, and in each of them, you can select the value from the specific filters, and then click anywhere outside this area sorry about that, to update these results and apply the filters and then update the results. So after applying these 3 filters, we are left with just 4 results, and they are all from Metabolomics Workbench. So if you click on the second one, that actually corresponds to the project of which the 2 studies ST000, 230, and 231 are part of. So on this details record page, we can find more information such as the project description and what is the sample source, what is the taxonomy, and what is the disease, which of course we partly saw in the results table on the previous slide as well. So, and then looking, and if you click on the project URL here, shown here, it will take you to the actual project page on the Metabolomics Workbench website where you can see the two studies are listed. So the purpose of this is that one could have first searched on the DRC CFDE portal as well and potentially identify this study to further follow up. Then we looked at, we searched for some of the genes that with which we started to begin with. So in this case, we essentially perform that main search as the liver cancer Homo sapiens. And then in that, there is a genes filter. In that one, you select the genes as the filter, and then you will get some results from the links or DCC, where they have information related to experiments in cell lines corresponding to the disease carcinoma, sample source liver and taxonomy Homo sapiens. So what we see here is that on the data portal, we can find a rich set of information which we can further explore. And in some cases, we will be able to download data files and we can further analyze the data files using suitable tools. So with this, I would like to conclude the demonstration. So essentially, starting with the key genes related to the disease of interest, a user can use MedGene to find the relevant metabolites and Metabolomics Workbench studies. Analyze these studies on Metabolomics Workbench using the tools available there, and then interpret the results using various tools, including such as pie chart, volcano plot, clustering, classification-related analysis, and the METLP pipeline. One can go to the CFDE portal and find more information related to the disease terms, key genes, and metabolites. So these tools essentially may help in hypothesis generation and further research by providing pointers in the right direction based on the results and interpretations they are able to provide. And mainly at the CFD data portal, one has the ability to download actual data files related to these searches we are looking for, and then carry out analysis. So with this, I conclude the presentation. And if you encounter any questions when you try some of these tools, then please contact the respective researchers listed here, depending upon what topic it is related to. And usually for each of these resources, we have a contact us link, or if there is a corresponding GitHub page, then on that repository, you can raise any issues or ask questions by looking at the issues tab. So with that, I thank you for your attention. Thank you for coming to the last day of the workshop, and we will be happy to answer any questions. Let me share my screen. So do you see this slide where this workflow is shown in a very high-level schematic? So this is one example that you start with a list of genes, and then you can follow different routes. For example, identify the studies and apply the analysis tools that are available at Metabolomics Workbench, and there are several of them. So as such, each of those as could be part of a more extended workflow, and MedGene analysis and METNP, those are examples. Then you can go to the CFDE portal and perform the search. Now, of course, this is a workflow at a very high level. If you want to construct a detailed workflow, for that, for example, if you look at the presentation I gave yesterday on the workflow builder tool, that is actually a tool for constructing a workflow based on all the tools that are available in the tool set. So if there is interest, I can actually go and bring up that presentation and then walk through that very briefly. So let me do that. So here is an example of workflow. Of course, in this one, for demonstration purposes, this workflow was prepared in PowerPoint for readability. And it is not necessary that all the components from this be available in the workflow builder tool, but nevertheless, most of them are. In fact, in this case, I believe all of them are available there. Now, in the actual workflow builder tool itself, as you saw yesterday, you could use the building blocks, for example, different tools from here and the starting points like nodes or gene input or gene set input and so on, and construct your workflow. Now, what workflow you are interested in developing depends on what research question you want to try to answer, right? So that largely depends on the type of study you want to conduct. But the way this workflow builder tool can help you is, once you decide what is your starting point, for example, whether it's a gene input or gene set or metabolite input, here if you can filter it on the left side, and then it will show you only the transformations which are applicable for that type of input. So in that sense, it guides you that what are the analysis you can apply on those types of input, and that applies for every step at every point in the workflow. I think as you saw, I can take some example. For example, when I am at this point here, when I click on this plus sign, you will essentially see that these are the only transformations that can be applied on that type of data input. For example, on this you can apply enrichment analysis. Going further in that workflow, I think I did not show the individual steps, but not for all, but for some I have showed. For example, here when I click on this, it tells me these are the analyses that you can apply on that. So that way the workflow builder tool can help you. It will not construct the workflow for you, but directly, but it can help you. In fact, it's a work in progress and it's a learning process, but this tool also learns what are the types of workflows that users have constructed and what is the corresponding summary for that. And based on that, they already have some ability that through a essentially AI-based assistant, where you can type or speak the question that you are trying to address, type out or just speak a little bit about your research study, that this is what you want to accomplish. And based on the learning it has done so far, it will try to come up with a tentative workflow, which you can further modify. So I did not demonstrate that feature in my presentation yesterday, but the core team which developed this app with in collaboration with the other 4 data coordinating centers, they do have two videos from a recent workshop that they had conducted, where they show how some of those features can be used. So please watch those videos, and those videos are actually available if you go to the GitHub page for Playbook Workflow Builder. I can put these links in the chat again. Then you can go through all the documentation there to understand better how you can use this workflow builder to the fullest extent for your work. Okay, while I'm going to post it in the chat, I'm going to start sharing.
So there's a follow-up from Suvita. It says, yes, so using the tool, we'll be able to understand the availability of the data, and so that accordingly we can plan our work design. Yeah, basically the CFDE data portal is designed for discovery, access, analysis, and finally generation of hypothesis. So the Playbook Workflow Builder is also integrated with the data resource portal, which is going to be available. I mean, that aspect is going to be available soon. So right now what we demonstrated was how to discover data and download data from the CFDE data portal. On the other hand, if you have your own data and you want to apply Playbook Workflow Builder, even that is possible, as Mano mentioned. Mano, if you want to add anything more, please feel free to add.
No, I think you covered it well. So in the Workflow Builder tool, there are several types of nodes. Let me share my screen again. Okay. Do you see the slide where it says Workflow Builder, Playbook Workflow Builder at the title of the slide? So here, as you can see, there are different types of node: gene input, gene set input, and then there are node for inputting, uploading your data matrix, whether it's related to genes person data or metabolite data or any other type of data matrix. Then depending upon the context, a subset of tools will show up here, such as if it is a data matrix, then you can apply principal component analysis or UMAP analysis, or you normalize that data matrix. Or you can compute you can apply other computations on that. And the tool set that we see here, it's an expanding set of tools. And users are also welcome if they have their own tools written in preferably in Python or TypeScript/JavaScript type of language. Then you will be able to integrate those tools. You can create your own modules following the guidelines, detailed guidelines provided at the GitHub page. And the second video in that set of two videos, that is especially dedicated to help users how they can develop their own modules in this and integrate that so that it becomes part of this workflow builder tool, so that of course it will be useful to them, and eventually it will be useful to the community as well once it is integrated into this tool set.